It's a Spotify podcast.
You pushed me down, thought I'd stay on the ground, counting me.
The treatment did not exist when the patient was born. What happens when the patient is the clinical trial? The treatment did not exist when the patient was born. There was no bottle waiting in a pharmacy, no approved drug with a familiar name, no clinical trial recruiting children with the same condition. There cannot be. The disease was extraordinary, rare. The mutation was specific, and the patient was getting sicker. So the team of scientists and physicians attempting something that sounded less like conventional medicine and more like emergency engineering. They studied the child's genetic code. They identified the error. They designated a molecular tool identified to correct it. They tested that tool. They manufactured it. They sought emergency regulatory permission. Then they infused it into the patient. Not a medicine selected for one child. A medicine created for one child. In 2025, an infant named KJ Moldon became the first known patient to receive a personalized gene editing treatment designed around a particular disease-causing genetic variant. The treatment used a form of CRISPR known as base editing and was delivered into his body by using a lipid nanoparentese. The case was extraordinary. But the deeper question is even more important. Was this a heroic exception? Or was it an early prototype for an entirely different model of medicine? Because modern drug development is built around a basic assumption one product should treat many patients. But what happens when the patient is only the person in the world with precisely that genetic error? What happens when a medicine cannot be mass produced because the disease itself is almost unique? And how do you prove that that treatment works when the clinical trial contains one human being? Strike that. When the clinical trial contains one human being. Welcome to another rabbit hole where biology meets regulation, economics, ethics, manufacturing, and meaning of medical evidence itself. This is the story of medicine for one. The patient called ETA shortly after KJ was born. Physicians discovered that something was terribly wrong. The patient called ETA shortly after KJ was born. Physicians discovered that something was terribly wrong. He had carbophosphate synthase 1 deficiency. This is the story of medicine for one. The patient called ETA shortly after KJ was born. Physicians discovered that something was terribly wrong. He had carboxyl phosphate synthase 1 deficiency, usually shortened to CPS1 deficiency. CPS1 is an enzyme used in the urea cycle, the biological process that helps the body remove nitrogen waste. When proteins are broken down, nitrogen is produced. The body normally converts that nitrogen into urea, which can then be excreted. But when the cycle fails, ammonia can accumulate in the blood. Ammonia is toxic, especially to the brain. Severe elevations can cause vomiting, lethargy, seizures, brain injury. Some some can cause death. KJ's disease was caused by mutations affecting the CPS1 gene. The condition is estimated to occur in roughly 1 person out of 1.3 million. And severe neonatal cases have historically carried an estimated mortality or about 50% during early infancy. The available medical options were limited. Doctors could restrict protein intake. They could use medications that help remove excess nitrogen. They could carefully monitor ammonia. Ultimately, a liver transplant might provide a functional source of the missing enzyme. But transplanting an organ into a critically ill infant carries its own risks. And time mattered. The research team referred to KJ initially as patient ETA. There is something almost unsettling about that name. It sounds anonymous, clinical, protective. But behind it was a real child whose body could not safely process one of life's most basic activities. Eating protein. His parents were not deciding between several proven treatments. They were watching physicians manage a biological emergency while researchers attempted to invent another option. That is where the story changes. Because the team did not begin by asking which existing drug might help. They asked, can we rewrite the specific genetic letter causing this disease? A typo with consequences. DNA is often described as an instruction manual. That metaphor is useful, but incomplete DNA is not a single book stored safely on a shelf. It is a chemical information system copied across trillions of cells. Its instructions are written using 4 molecular letters: A, C, G, and T. A genetic mutation may involve a large missing section, a duplicated region, a rearrangement, or sometimes a change in a single letter. Case 9 is often compared with molecular scissors because it can cut both strands of DNA at a selected location. Base editing works differently. Rather than making a complete double-strand break, a base editor can chemically convert one DNA letter into another at a targeted site. The technique combines a modified CRISPR targeting system with an enzyme capable of changing a particular nucleotide. Think of it less as cutting a paragraph out of a book and more as correcting a typographical error without tearing the page. That sounds precise, but biology is not word processing. The editor must reach the correct cells. It must enter those cells. It must find the intended DNA sequence. It must make enough corrections to matter. It must avoid making harmful changes elsewhere. And it must not provoke a dangerous immune reaction. The researchers, therefore, needed more than a generic idea. They needed a complete delivery system. For KJ, this customized editor was packed inside lipid nanoparticles, tiny fat-based carriers related in broad concept to delivery systems used for some RNA medicines. Those particles were intended to carry the editing machinery to the liver cells. The liver mattered because CPS1 normally performs its function there. Correcting every cell was not necessary. The hope was that editing enough liver cells could restore enough enzyme activity to improve ammonia control. But before anyone knew whether that would happen, the team had to create a product that had never existed. And they had to do it while the patient was alive, vulnerable, and waiting. 6 months to build a medicine. Drug development is normally slow. Candidates are discovered, optimized, tested in cells, tested in animals. Manufacturing processes are developed. Regulatory submissions are prepared. Clinical trials proceeded through phases. Safety is monitored. Evidence accumulates. The process can take years, sometimes more than a decade. KJ didn't have a decade. The researchers began developing the customized therapy soon after his diagnosis. According to the clinical report, the treatment received regulatory authentication and was administered in 2 infusions when he was approximately 7 and 8 months old. The scientific achievement was not merely that the edited a gene. Researchers had edited genes before. The achievement was the workflow, diagnosis, genetic sequencing, editor design, laboratory testing, manufacturing, safety analysis, regulatory review, clinical delivery, all compressed around the needs of one patient. This is what makes the case potentially historic. A custom treatment had moved from genetic diagnosis to administration in roughly half a year. That does not mean future treatments can always be produced that quickly. It does not mean that process was inexpensive. And it does not mean that outcomes prove therapy will provide a permanent cure. But it did demonstrate that individualized editing programs can move through a real medical system, not merely succeed in a laboratory dish. The treatment was no longer theoretical. It entered a human body, and then everyone had to watch. What happened to KJ? After treatment, KJ was able to tolerate more dietary protein and required lower doses of nitrogen scavenging medication, according to the published clinical report, and NIH summarizes. He also survived infections that would ordinarily create serious metabolic stress for someone with CPS1 deficiency. Those observations were encouraging, but scientific honestly requires careful language. This was one patient. There was no control group. The child continued to receive other medical care. The precise proportion of liver cells successfully edited in the body could not simply be measured by removing large amounts of liver tissue. Long-term safety remained unknown. Long-term durability remained unknown. Researchers could not yet know whether the invention would eliminate the eventual need for a liver transplant. The treatment should therefore not be described as definitive proof that personalized gene editing cures CPS1 deficiency. The evidence supports a narrower but still remarkable conclusion. A customized base editing therapy was successfully designed, manufactured, authorized, and administered to one infant with early clinical changes consistent with therapeutic benefit and without the researchers reporting serious treatment regulated toxicity during the initial observation period. That is not a small conclusion, but it is different from saying CRISPR cured the baby. One is an evidence-based description. The other is a headline. And this distinction takes us into the first major complication. What does evidence mean when there can never be a conventional trial? Medicine was built for groups. Modern medical science has spent generations developing ways to protect patients from treatments that merely look promising. That history matters. For centuries, physicians relied heavily on personal observation, tradition, and authority. A respected doctor could claim a treatment worked because some patients improved. But patients improve for many reasons. Diseases fluctuate. Symptoms disappear temporarily. Other treatments contribute. Diagnosis can be wrong. Human memory is selective. Hope changes perception. And sometimes people recover despite the treatment, not because of it. Randomized controlled trials were developed to reduce those uncertainties. Patients are assigned to treatment and comparison groups. Researchers define outcomes in advance. Enough participants are enrolled to detect whether an apparent effect is more than a chance. Blinding can reduce expectation effects. Replication can reveal whether results survive outside one institution. The framework transformed medicine. It helped expose it. It identified harmful inventions, and it established an evidence standard stronger than expert intuition. But randomized trials depend on numbers. What happens when there are no numbers? An ultra-rare genetic condition might affect a few dozen people. A particular variant might appear in only one family. Some patients may be infants, others adults. Their disease's stages may differ. Their mutations may disrupt the same gene in different ways. Even assembling 10 comparable patients may be impossible. Now imagine that treatment itself changes for each mutation. One patient receives editor A, another receives editor B, a third receives editor C. They share a delivery platform and a biological strategy, but the molecular sequence is not identical. At that point, what exactly is the product being tested? Is the product each customized editor? Or is it the product, the platform used to design and deliver editors? That may sound like regulatory wordplay. It is not. The future of personalized medicine could depend on the answer. The platform is the product. Consider vaccines. Every year's influenza vaccine may contain different strains. The formulation changes, but the regulators do not treat each seasonal update as an entirely unrelated invention starting from zero. Manufactured methods, platform knowledge, and prior safety data can carry forward. Something similar occurred with the updated COVID-19 vaccines. The exact antigenetic target changed while important features of the underlying platform remain familiar. Individualized gene editing pushes this concept much further. The targeting sequence may change for every patient, but other elements might remain stable. The same lipid nanoparticle, the same base editing enzyme, the same manufacturing facility, the same quality control framework, the same methods for predicting off-target changes, the same laboratory assays, the same target organ, the same dosing logic. If those shared components can be validated, perhaps regulators do not need to treat each personalized editor as though scientists know nothing about it. The idea is known broadly as leveraging prior knowledge. In June of 2026, the U.S. Food and Drug Administration issued draft audience specifically addressing how prior knowledge might be used in developing human gene therapy products that incorporated genome editing. The document remains draft guidance, meaning it contains non-binding recommendations rather than a final enforceable rule. The FDA's movement did not begin there. In February of 2026, the agency introduced a draft plausible mechanism framework for individualized therapies targeting ultra-rare genetic conditions with known biological causes, rather than demanding the kind of large randomized evidence impossible in these populations. The proposed framework considers whether a treatment has a well-supported mechanism, reaches the relevant biological target, and produces objective evidence consistent with benefit. The agency also issued draft guidance on genome editing safety standards and broader development flexibility for cell and gene therapies during 2026. Taken together, these documents indicate that regulators are beginning to confront a new reality. Medicine may soon contain products that cannot be evaluated through traditional mass market development pathways. But here's where the story becomes difficult. Flexibility can save lives. Flexibility can also lower barriers designed to protect patients. And both statements can be true. The evidence problem. Imagine that your child has a fatal genetic disease. There is no approved therapy. Researchers believe they can design an editor that might correct the mutation. Animal studies look promising. Laboratory testing does not reveal obvious danger. But no person has ever received that exact molecular product. What level of evidence would you require? Now, change your position. You are no longer the parent. You are the regulator. Your decision will not affect only one family. It may establish a precedent for hundreds of future therapies. Approved too slowly, and a child may die with a plausible treatment remains in a freezer. Approved too quickly, and an inadequately tested editor could cause an irreversible harm. This is not a conflict between compassionate people and bureaucrats. It is a conflict between different kinds of protection. Protection from disease and protection from unproven treatment. The more desperate the situation, the more difficult it becomes to separate those protections, because desperation changes the psychology of evidence. A small chance may feel enormous when the alternative is death. An unknown risk may feel acceptable when the known risk is catastrophic. Parents may rationally accept uncertainty that would be unacceptable in a common, slowly progressing condition. But informed consequence becomes complicated when scientists themselves cannot quantify the risk. How do you explain the probability of a harmful, off-target edit when the exact therapy has never been used before? How do you explain the possibility of immune toxicity? How do you discuss risks that might appear 10 or 20 years later? And what does consequent mean when the patient is an infant? These are not arguments against personalized therapy. They are reason that the field requires usually rigorous honesty. Off target does not mean imaginary. Gene editors are designed to bind to particular DNA sequences, but biological systems contain repeated and similar sequences. An editor could potentially act somewhere unintended. These are known as off-target effects. Not every off-target change is dangerous. Some may occur in nomic regions with no meaningful consequences. Others could disrupt an important gene in the worst case. An unintended change could contribute to cancer or another serious condition. Researchers therefore use computational prediction, cell-based testing, sequencing, and animal studies to search for unwanted edits. But no test can guarantee that every possible long-term risk has been eliminated. There can also be unintended changes at the intended site. An editor might produce the desired conversion in some cells while causing different changes in others. Delivery adds another layer. Lipid nanoparticles can prefer intentionally. Delivery adds another layer. Lipid nanoparticles can prefer intentionally. Naomi, strike that. In some cells while causing different changes in others. Delivery adds another layer. Lipid nanoparticles can preferentially reach the liver, but distribution is not perfectly exclusive. The immune system may react to delivery materials or editing proteins. Dose matters. Timing matters. The patient's condition matters. None of this means gnome editing is recklessly uncontrollable. It means precision exists on a spectrum. Gene editing sounds like a perfect cursor placed over a single letter. The reality is closer to biochemical probability engineering. Researchers can design, test, improve the odds. They cannot turn a living organism into completely predictable document. That is why long-term monitoring will be essential. The first months after treatment can reveal acute toxicity. They cannot answer what happens over decades. One success can mislead us. Human beings naturally remember dramatic cases: a sick child, a race against time, a custom treatment, and apparent improvement. The story has everything required to become a symbol, and symbols can distract judgment in both directions. Supporters may treat KJ's case as a proof that personalized gene editing has arrived. Critics may treat every uncertainty as proof that the technology is premature. Both reactions exceed the evidence. The case demonstrates reasonability. It does not establish scalability. It demonstrates a plausible clinical benefit. It does not establish universal safety. It demonstrates that one exceptional team, supported by major institutions and specialized collaborators, could create one treatment under urgent circumstances. It does not establish that ordinary hospitals can repeat the process for thousands of patients. This is where the field must resist what might be called prototype confusion. A prototype proves something can happen. A system proves it can happen repeatedly. Medicine, for one, has produced a prototype. The next challenge is building the system. And systems are not made of science alone. They are made of institutions, money, manufacturing, rules, expertise, access, and time. The economics of the impossible. Traditional pharmaceutical economics favors scale. Drug development is expensive. A company invests in discovery trials, manufacturing, regulatory submissions, and marketing. The cost can be recovered if enough patients buy the product. That model works best when treatment serves a large market. It becomes increasingly difficult as the patient population shrinks. Rare diseases medicines can command extremely high prices, particularly because development costs are spread across fewer people. Gene therapies have already reached prices in the millions of dollars per treatment. Now take the logic to its extreme. What is the price of a medicine made for one person? If every customized editor requires separate design, testing, manufacturing, and regulatory review, the economics become nearly impossible. A company cannot build a conventional business around spending millions to create a product it may sell once. This is why the platform concept matters so much. The individualized component must become only a portion of the work. Researchers need reusable editor systems, reusable delivery vehicles, automated design pipelines, standardized safety assays, shared manufacturing, regulatory templates, databases of prior results, and perhaps libraries of editors covering many possible mutations. The goal is not literally to create every therapy from scratch. The goal is to make a customized, reasonable, changing one validated component inside an established system. Nature commutators, all right, and perhaps libraries of editors covering many possible mutations. The goal is not literally to create every therapy from scratch. The goal is to make customized, resemble changing one validated component inside an established system. Nature commentators argued in 2026 that fresh approaches to testing and grouping personalized CRISPR therapies could potentially make treatment economically viable for thousands of patients with rare genetic diseases. That is a proposed pathway, not evidence that widespread economical delivery has already been achieved. The distinction matters. The science may be moving faster than a business model. A therapy can biologically be possible and economically unavailable. Modern medicine already contains many examples of this gap. Personalized gene editing could widen it dramatically. Who deserves? Suppose a platform can produce individualized therapies. Which patients receive them? The sickest? The youngest? Those with mutations easiest to edit? Those with diseases that affect the liver, where lipid nanoparticles currently offer a practical delivery route? Those living near elite research hospitals? Those whose families can raise public attention? Those whose insurers agree to pay? Those born in wealthy countries? Scientific feasibility does not distribute itself equally. The first patients are likely to be selected partly by biology. Some organs are easier to reach than others. The liver is comparatively accessible to certain delivery systems. Blood-forming stem cells can be removed, edited outside the body, and reinfused. The brain is much harder. Muscle is enormous and widely distributed. The lungs present different barriers. A child with an edible liver disorder may therefore have a path that does not exist for another child with an equally severe neurological disease. That difference is not moral. It is technological. But technology can become inequality. And then the inequality becomes political. If a health system can spend several $1 million to create one personalized therapy, what other care goes unfunded? Is it fair to compare a one-time potentially curatively treatment with decades of hospitalizations and supportive care? Should cost effectiveness calculations apply to the same way ultra-rare childhood diseases? Should governments treat individualized genetic medicines as public infrastructure? Should nonprofit consortia develop them when commercial markets are too small? Should platforms create with public research funding remain openly available? Or will future of medicine for one belong primarily to private companies controlling editor patients, delivery technology, manufacturing, and data? These questions are not side issues. They may decide whether personalized gene editing becomes medicine or merely a series of extraordinary demonstrations. The data commons. Every customized therapy could generate knowledge useful for the next one. Which editor were designed effectively? Which sequences feature predicted off-target activity? Which doses reached enough cells? Which immune responses occurred? Which manufactured methods worked? Which changes remain stable? The information can make future treatments faster and safer, but only if it is shared. A fragmented system would force each institution to relearn the same lessons. Companies might treat design information as proprietary. Hospitals might store incompatible data. Privacy rules could limit access. Small patient numbers could make even de-identified records recognizable. Families may want to contribute data to help others, but fear misuse of their child's genome. The success of medicine for one may therefore depend on creating a trusted knowledge commons, not a public database containing children's names and complete gnomes. A carefully governed system through which researchers can learn from previous therapies while protecting patients. This creates an unusual tension. Personalized medicine becomes more individual at the point of treatment, but more collective at the point of knowledge. One person's therapy may depend on data generated by hundreds of previous patients, and every patient treated becomes part of the evidence base for someone not yet born. The history beneath personalized medicine. The idea of tailoring treatment to the individual is not new. Physicians have always adjusted medicines by age, weight, symptoms, and responses. Blood types made transfusion more individualized. Tumor makers began dividing cancers into biological subgroups, pharmacogenetic and pharmacogenomic. Naomi, strike that. Always adjusted medications by age, weight, symptoms, and responses. Blood types made transfusion more individualized. Tumor markers began dividing cancers into biological subgroups. Pharmacogenomics has revealed that genetic differences can alter how people metabolize drugs. Targeted cancer therapies tie treatment to specific mutations. CAR T-cell therapy involved modifying a patient's own immune cells. Gene therapy attributed to provide working genetic instructions. CRISPR introduced more direct genome modification. What is new is the degree of customization. Personalized medicine once meant selecting the right existing treatment for a patient. Then it meant selecting treatment based on molecular subtype. Now it may mean manufacturing a molecular intervention whose sequence changes because of one patient's genome. That is not merely another step. Naomi, strike that. Whose sequence changes because of one patient's genome. That is not merely another step along the continuum. It changes the relationship between diagnosis and treatment. In conventional medicine, diagnosis identifies the disease. Then the doctor chooses a product. In individualized genetic medicine, diagnosis may become part of the manufacturing. The patient's mutation helps define the product itself. The medical record becomes a redesigned specification, and that raises a philosophical question. When a therapy exists only because of one person's genome, where does the patient and the product begin? What counts as a cure? There is a temptation to frame gene editing as the final answer to genetic disease. Find the mutation, correct it, cure the patient. Sometimes biology may work that way, but often it will not. A disease-causing mutation may begin damaging tissues before birth. Correcting the mutation later may stop further injury without reversing what has already occurred. Some conditions involve many genes. Others involve a combination of genetic and environmental factors. Some target cells divided rapidly, diluting the effect. Other tissues are difficult to reach. A treatment may improve function without restoring normal biology. Editing may be incomplete. Benefits may fade. A patient may still need medication. Calling every genetic intervention a cure risks obscuring these realities. KJ's treatment may reduce the biological consequences of CPS1 deficiency. It may eventually remove the need for some forms of support. It may provide durable benefit. But the evidence must mature before anyone can know the full answer. Perhaps cure is not always a single event. Perhaps it is a threshold. Enough corrected cells, enough enzyme activity, enough metabolic stability, enough reduction in risk before child to strike that. Enough corrected cells, enough enzyme activity, enough metabolic stability, enough reduction in risk for a child to live a life that was previously impossible. Medicine often works in gradients, even when headlines prefer absolutes. There are at least 3 plausible futures for medicine. For one, the conservative future. Personalized gene editing remains rare. A small number of elite academic centers create treatments for exceptional patients with life-threatening conditions and especially suitable mutations. Regulators allow case-specific flexibility, but design and manufacturing remain expensive. Most ultra-rare diseases continue to lack treatments. KJ's case became historically important because the model spread slowly. In this future, individualized editing resembles organ transplantation in its early decades. Transformative, real, but limited to specialized institutions and carefully selected patients. The platform future. Editor design becomes increasingly automated. Off-target prediction improves. Manufacturing becomes standardized. Validated delivery systems can be reused. Regulators accept platforms evidence. Groups of patients with different mutations in the same gene can be evaluated under shared protocols. Newborn genomic screening identifies candidates before irreversible damages occur. Hospitals submit genetic information, coordinated development networks. A customized therapy can be produced in weeks or months at a fraction of today's cost. In this future, medicine for one becomes medicine for thousands. Not one identical drug for thousands of patients. Thousands of related medicines produced by a shared system. The disruption future. A serious adverse event changes public and regulatory attitudes. An editor causes an unexpected mutation. A patient develops cancer. A manufacturing error harms someone. A company collapses after pricing controversy. Insurance systems refuse coverage. Patient disputes prevent platform sharing. Or another technology, perhaps RNA editing, epigenetic editing, or improved gene replacement, proves safer and easier to standardize. In this future, personalized DNA editing remains scientifically plausible, but loses momentum. The lesson of medical history is that breakthroughs rarely follow a straight line. Technologies advance, then complications appear. Standards tighten, methods change, expectations adjust. The future will probably contain elements of all three scenarios. The child and the system. Return to the beginning. A newborn. A genetic error. A treatment that did not exist. At first, the story seems to be about CRISPR, but CRISPR is only one layer. The real system includes newborn diagnosis, genomic sequencing, computational design, molecular biology, animal testing, manufacturing, quality control, hospital care, regulation, ethics, insurance, data sharing, intellectual property, and the willingness of a family to enter a territory where medicine has almost no map. The editor may be personalized. The achievement was collective scientists who developed base editing, researchers who created delivery systems, physicians who recognized the disease, technicians who manufactured the product, regulators who reviewed the evidence, nurses who monitored the patient, public institutions that funded foundational work, and parents who accepted an uncertainty no parent should ever have to face. That is why medicine for one is slightly misleading. The treatment may be made for one person, but creating it requires an entire civilization of accumulated knowledge. The bigger question: for most of medical history, rarity was a disadvantage. And few people shared your disease, fewer doctors recognized it. Fewer researchers studied it. Fewer companies invested in it. Fewer treatments were developed. Personalized gene editing raises the possibility that rarity may one day become less decisive. A disease might not require a large market of scientists that can rapidly modify an existing therapeutic platform. But that future does not arrive merely because a molecular tool exists. One patient's success does not automatically become a public health system. To reach that future, society must solve a series of problems that are arguably harder than editing one DNA letter. How much evidence is enough? Which risks are acceptable? What can be learned from one patient? What knowledge can be reused? Who owns the platform? Who pays for the treatment? Which diseases are prioritized? Naomi, strike that. Which diseases are prioritized? How long-term effects tracked? How do we prevent access from depending on wealth, geography, or publicity? And perhaps the hardest question of all: can medicine become radically individualized without abandoning the collective evidence standards that made modern medicine trustworthy? That is the paradox. The future may require treatments designed for individuals, but reliable knowledge is created through accumulation, comparison, transparency, replication, and shared experience. The patient may be one. The evidence cannot remain one forever. KJ's story does not prove that we have entered an age made to order cures. It proves something narrower and perhaps more important. A group of people looked at a disease affecting almost nobody and refused to accept that almost nobody meant nobody would try. They transformed a genetic diagnosis into a molecular design. They transformed a design into medicine. And they placed that medicine into the body of a person whom it had been created. Now, the responsibility shifts to the rest of us. Not to celebrate without skepticism, not to fear without evidence, but to decide what kind of system should grow around this possibility. What evidence changed your thinking? Would you accept an individualized gene editing therapy if the alternative were a fatal disease? How much uncertainty would be acceptable? Should one early success justify a faster regulatory pathway? Who should pay for medicine that may treat only one person? Should the underlying platforms be treated as commercial products, public infrastructure, or some combination of both? What protections would make you trust the system? And when the next child is born with a mutation no therapy has ever targeted, will medicine tell that family the disease is too rare? Or will rarity itself become the reason to design something new? If today's episode made you think, don't stop here. Read the research, study history, examine original documents, listen to the experts, listen to the critics, listen to the people who disagree with you, question the sensational claims, question official explanations, Question the headlines. Question the experts. Question your assumptions. And question mine. Because Rabbit Holes Deep Dives is not about handing you a conclusion. It's about following the evidence, even when the evidence leads somewhere uncomfortable. What evidence changed your thinking? Which claim deserves more investigation? What did we miss? Which assumption should we challenge next? And what rabbit hole should we go down next? Share this episode with someone willing to examine the evidence. Join the conversation. Challenge the ideas respectfully. Keep asking questions, because every answer opens another question, and every question leads us further down the rabbit hole. Say it in the categories below. I read every one. My name is Leroy Nellis, and this is Resilience Radio. Godspeed, stand for the Constitution, and never surrender your ability to think for yourself. God bless, and good night. Hey, everybody. I wanted to sit down with you today and talk about something we've been working on behind the scenes for quite a while now. First, I want to thank you. Whether you've been here from the beginning or you just found the channel recently, I want you to know that I genuinely appreciate every single one of you. Every comment, every conversation, every suggestion, every word of encouragement has helped shape what Resilience Radio is becoming. This isn't just my channel anymore. It's about a community. And because of that, I wanted you to hear these changes directly from me. Over the last several months, we've been doing something a little unusual. We've been studying our analytics almost obsessively. Not just YouTube analytics, but the analytics coming from Naomi, the AI production system we've been building from the ground up. We've been asking questions like, what videos do people come here for? What keeps people watching? What topics naturally fit together? And what happens when we mix very little types of content on the same channel? After looking at thousands of data points, we came to one conclusion. It's time to let each series become a very best version of itself. So today, I'm excited to announce that Rabbit Holes is becoming its own dedicated YouTube channel. Rabbit Holes deserves its own home. It's where we dive into mysteries, history, unsolved questions, and rabbit holes. Strange events, forgotten stories, and all of those fascinating topics that make us lose track of time. By giving Rabbit Holes its own channel, YouTube can better understand who those videos are for. Our AI can learn from much cleaner data, and we can build something truly special for people who love those deep dives. Resilience Radio will continue doing what has always meant to do, help people understand themselves through psychology,
You pushed me down, thought I'd stay on the ground, counting me out like the numbers didn't matter. But pressure builds diamonds in the dark, and I've been sharpening my edge in the chatter. Every scar turned into strategy, every setback fueled my clarity. I took the hits, learned the game, turned my losses into flame. Now I'm coming back strong, and you'll remember my name. I'm rising through the fire.